Welcome to this explainer. Today, we're taking a completely mind-blowing microscopic factory tour to discover exactly how your body creates the immense energy required to literally keep you alive. We're going to be looking at the grand finale of energy metabolism. It's a process called oxidative phosphorylation, and let me tell you, it is, without a doubt, a masterclass in nanoscale engineering. So let's start with a really fundamental question. You eat a sandwich, right? But how exactly does your biological machinery transform that sandwich into actionable mechanical power? Sure, you consume carbohydrates, fats, and amino acids, but those molecules can't just directly flex a muscle or fire a neuron on their own. Their energy has to be converted first into a universal cellular currency, a molecule we call ATP. Now get this, 70%. That astonishing number represents the thermodynamic efficiency of your cellular engines under normal physiological conditions. To put that into perspective, a typical automobile engine operates at roughly 30% efficiency. Your cellular machinery is somehow managing to capture about 70% of the available free energy from your fuel. How on earth does biology achieve something that completely and totally eclipses our own human engineering? Let's dive into Section 1, the cellular power plant, and explore the anatomy of this factory floor. To really grasp that crazy 70% efficiency, we have to look at the physical factory floor itself, the mitochondrion. People always call it the powerhouse of the cell, and for good reason. This is where all the oxidative steps from breaking down your food finally converge. What's really crucial here is the structure. You have this highly folded inner membrane called cristae, and those folds are there to maximize the surface area, basically packing in as much power-generating machinery as physically possible. While the outer membrane lets small molecules pass through pretty easily, that inner membrane is strictly impermeable to almost everything, especially protons. This creates an isolated inner compartment, the matrix, and that separation is absolutely vital for everything that's about to happen. And to grasp the sheer, unbelievable scale of this operation, a single liver mitochondrion contains over 10,000 of these microscopic electron transfer systems densely packed all over that folded inner surface. 10,000. And in tissues with super high energy demands, like your heart muscle, they are packed even more tightly together. It's an incredibly crowded, buzzing factory floor. Moving on to Section 2, the electron assembly line, and how fuel actually moves through the system. So how exactly is fuel processed on this microscopic assembly line? Well, fuel arrives in the form of electrons, carried by molecules like NADH. They reach that inner membrane and embark on a very precise step-by-step journey. They're passed through a series of massive protein structures known as complexes I, 3, and 4. And between these giant stations, you have highly mobile little shuttles like ubiquinone, also known as coenzyme Q, and cytochrome C, that ferry the electrons forward. Finally, at the end of the line, the electrons are handed off to the very oxygen you breathe, reducing it safely to form plain old water. And looking at the actual structures of these complexes, it proves that this isn't just some random chemical soup floating around. We are looking at actual physical heavy machinery passing electrons down like stepping stones. Complex 3 and Complex 4, for example, are packed with iron-sulfur clusters and specialized heme groups that carefully, methodically pull the electrons downhill to lower and lower energy states. It's molecular engineering at its absolute finest. Next up is Section 3. Building the proton dam, which brought about a total paradigm shift in biology. The key thing to understand is that the energy released from moving all those electrons isn't just wasted as heat, it's harnessed to build an incredible biological dam. As electrons flow spontaneously through the complexes, the energy physically forces the machinery to change shape. And this brings us to Peter Mitchell's revolutionary 1961 Chemiosmotic theory. He realized that as the electrons flow down the line, protons are physically being pumped out of the matrix and into the inner membrane space to store that energy. I can't overstate this. This was a massive paradigm shift. It elevated oxidative phosphorylation from just another basic metabolic pathway to a true marvel of nanotechnology. Before Mitchell, scientists were hunting for some kind of direct, chemical, high-energy intermediate. Discovering that the energy was actually stored spatially as a physical and electrical gradient across a membrane, well, it completely changed how we understand biology. By relentlessly pumping those protons into that intermembrane space, the cell is building up immense electrochemical pressure. It acts exactly like water building up behind a massive hydroelectric dam. We call this the Proton-Motive Force. Because protons have a positive charge, pumping them out creates a dual gradient. First, you get a chemical gradient, where the inside matrix becomes more alkaline, and second, you get an electrical gradient because the outside literally becomes more positively charged than the inside. Which brings us to section 4, the ATP turbine, where we finally open the floodgates. Okay, this is the absolute climax of our factory tour. All those protons are desperate, really desperate, to flow back across that impermeable membrane to balance out the charge and concentration. And the mitochondrion provides exactly one way back in. As the protons rush back through, we get to witness something called rotational catalysis. There's an enzyme called ATP synthase that literally spins like a mechanical turbine as protons rush through its base, physically rotating a ring of protein subunits. This rotary engine perfectly couples that massive rush of protons to the mechanical release of newly minted ATP molecules, ready to go power your cell. The rotation forces the top part of the enzyme to change shape. And here's the craziest part. Forming the ATP molecule itself actually requires very little energy. The massive, rushing energy from the proton flow is primarily used just to physically push the newly synthesized ATP off the enzyme so the cycle can start all over again. But what happens when things go wrong? Let's look at section 5, short-circuiting the system, or when the factory leaks. So what happens if this tightly controlled proton dam springs a leak? Remember, this whole system relies entirely on the inner membrane being totally impermeable to protons, so they are literally forced through that ATP turbine. We call a leak uncoupling. If protons find a shortcut back inside, bypassing the turbine, the electron transport chain starts running frantically to rebuild the dam. It consumes oxygen, it burns through your fuel, but absolutely zero ATP gets made. This short circuit essentially turns your cellular factory into a pure heat generator. Now, this can be artificially induced by dangerous chemical uncouplers like DNP, which literally ferry protons across the membrane, and that can be deadly. But biology actually uses this deliberately too. In brown fat tissue, specialized uncoupling proteins allow protons to purposefully bypass the turbine just to generate heat. It's a vital survival mechanism for things like hibernating animals and newborn babies to maintain their body temperature. Finally, let's wrap up with section 6, the vital importance of mitochondrial integrity. Maintaining the integrity of these microscopic power plants is absolutely fundamentally essential for your survival. When they're functioning correctly, they are a total marvel of thermodynamic efficiency. Just look back at this comparison. Extracting energy from fuel without any combustion, using physical nanomachinery to pump a proton dam, at 70% efficiency, it vastly outperforms our best automobile engines. The stakes here are incredibly high. A single molecule of glucose yields roughly 32 ATP through this system. When this assembly line breaks down, whether the membrane gets leaky or the turbines degrade, tissues with high energy demands, like your brain and your muscles, they suffer first. In fact, a loss of integrity here is strongly linked to whole body aging and plays a central role in neurodegenerative conditions, diabetes, and obesity. And that leaves us with a pretty profound final thought about our own biological machinery. If the gradual loss of this system drives aging and disease, what can we do to one day keep these cellular power plants running at peak efficiency forever? By understanding oxidative phosphorylation, we're not just learning about some textbook biology, we are literally looking at the fundamental spark of life itself. Thanks for coming along on this factory tour.
Thank you for watching. See you in the next one. Stay tuned.